Hello, my name is Manuela, and I'm going to present you an article about phagosomal environment that the topic phagosomal environment determines antigen degradation and presentation. So this article is from 2002 from Cell, and it says that NOX2 controls phagosomal pH to regulate antigen processing during cross-presentation by dendritic cells. So we have to clarify what cross-presentation is. Cross-presentation has two theories. One that cross-presentation cells that some dendritic cell subsets have special antigen processing machinery that allows the exogenous antigens to go to class 1 AMHC. And the others say that antigen can be directed to endosome where class 1 is loaded with the antigen. So the importance of this process is that besides AMH2 pathway presenting antigens to CD4 or helper cells, disease can put an quick action kind of presentation when the exogenous antigens go to phagosome or are processed in endosomes to be presented as MHC class 1 so CD8 cytotoxic cells can recognize it and do their job. So this can be bad for the decedent cells because it's a risky thing. They could be targets of lysis themselves. So I'm gonna say that in the beginning of most cytotoxic immune response, disease must present proteolytic peptides derived from fat pathogens or infected cells to naive CD8 lymphocytes. Phagocytosis antigens are present to CD8 lymphocytes through a process called cross-presentation. It requires antigen transit to endosomes or phagosomes. These peptides are then translocated into the lumen of the endoplasmatic reticulum or back to specialized mixed phagosome ER compartments. This initial partial degradation of antigens must be tight controlled as it could destroy potential peptides for T cells. In phagocytic cells, protein degradation is mediated by a large family of proteases, lysosomal proteases. Hello, my name is Manuela, and I'm going to present you the following article on phagosome environment, determination of antigen degradation presentation. This article is called NOX2 controls phagosome pH to regulate antigen processing during cross-presentation by dendritic cells. It's an article published on Cell in the year of 2006. So to initiate most cytotoxic immune response, disease must present proteolytic peptides derived from pathogens or infected cells to naive CD8 T lymphocytes. Phagocytosis antigens are present to CD8 through a process called cross-presentation. Cross-presentation requires antigen transit through endosomes or phagosomes and eventually partial proteolysis before transfer to the cytosol and degradation by proteasome into amino acids. These peptides are then translocated into the endoplasmatic reticulum or back into specialized mix of ER phagosome compartments and loaded into MHC1 molecules. So this process have two postulates that disease have a special antigen processing machinery, and the antigen can be directly derived to the endosome where class 1 MHC is loaded with it. So it's a good process because it permits quick action of CD8 cytotoxic cells, the recognition. And it's a disadvantage because Disease could be targets of lysis themselves. This initial partial degradation of antigens must be tightly controlled, as it could destroy potential peptides for T-cell recognition. So it's a cross-presentation. It's the process of metabolize antigens in presenting them in MHC1 molecule that will facilitate the fast action in the adaptative immune response, but it's risky thing because they could be target of lysis themselves. So basically, in phagocytic cells, protein degradation is mediated by a large family of proteases, often referred as lysosomal proteases. It's a complex of enzymes. Most lysosomal proteases have optimum proteolytic activity between pH 5.5 and 6.5. And basically the process consists in phagosome fusion with early and late endosomes and then lysosomes, acquiring progressively both the acidification machinery and the lysosome proteases from the endocytic pathway. So these enzymes are important for the degradation, and the acidification or alkalinization determines their activity and the level of degradation of antigens. So in macrophage, phagosomes acidify very efficiently because the main goal of macrophage is effective microbial toxicity, microbial killing. Protein degradation it's very effective. Acidification is mainly but not uniquely mediated by the vacuolar ATPs, which translocate protons. It's like a pump from the cytosol into the lumen of endosomes, lysosomes, and phagosomes. Protons can also get in phagosomes through other conditioned channels, such as voltage-gated proton channels. So the acidification is mediated by the activity of these ATPs. In neutrophils, there is another larger protein complex, the NADPH oxidase NOX2, that mediates the transfer of electrons across endocytic and plasma membranes and also influences the phagosomal pH. So it's a multi-component enzyme, unassembled and inactive in resting cells, but assembles at phagosomal plasma membrane in a stimulus-dependent manner. Oxidase subunits include cytosolic proteins such as P45, P65, P40, and HAC1 and HAC2. Only here represent only HAC2. An integral membrane heterodimer cytochrome B558 composed by GP91 FOX and GP22 FOX. The production of ROS in neutrophils consumes very important amounts of protons, causing transient but strong alkalinization, pH 8, of the phagosome lumen during the oxidative burst. But as it ends, the ROS production stops, phagosome acidification resumes, and the pH drops in 30 minutes. So disease also expresses NOX2, but at really low levels, around 5%. The very low levels of ROS production in disease suggest that NOX2 may not be involved in microbe killing, since the particular main goal of a professional presenting antigen cell is most related to the initiation of adaptive immune response and not to microbe toxicity. So disease have developed specific means of control of endocytic functions that allow efficient antigen presentation. These specializations of endocytic pathway include the regulation transport of MHC molecules, the transport of antigen from endosomes and phagosomes into the cytosol, and the recruitment of ER proteins to phagosomes. So disease endocytic pathway can be considered as specialized antigen presentation compartments rather than purely degradative organelles. Knowing this, the group decided to investigate. The main goal of the group was to investigate these kinds of specialization of the endocytic compartments in disease and see if they are genetically indispensable. They are looking for evidence in the time there weren't genetically evidence of these specializations being indispensable for antigen presentation to T cells. So first, they decided to measure phagosomal pH in disease accurately. So they set up a phagosomal pH measuring by flow cytometry, fluorescence activated cell sorts of FACS. So the lattice beads were coated with pH sensitive and pH insensitive fluorescence dyes. After different times of phagocytosis of the beads, the fluorescence intensity was quantified using facts. So the ratio in fluorescence intensity between the two dyes affects the pH in phagosomal environment. As you can see in Figure 1, this is an active system for phagosomal alkalinization. Figure 8 shows that the pH in disease compared to a lineage of macrophage is much higher, it's much alkaline, it's less acidic than macrophage. So we can see after 20 minutes of phagocytosis pulse and 30 minutes of incubation, the fluorescence intensity is much higher in disease than macrophages. And you can see in Figure B, 1b, that in contrast, pH in DC phagosomes is much higher. It's much higher, reaching 7.5, around 7.5, than bone marrow derived macrophage and the raw cell line of macrophages that are acid and acidify very fast. A similar alkaline phagosome pH was also found in fresh isolates. Okay, therefore the pH in disease is alkaline and does not acidify significantly in the first 3 hours of phagocytosis. Okay, so the group hypothesized that if such a colonization process existed in disease, should become more evident upon blockage of the VETPs present in disease phagosomes. So the group analyzed disease phagosome pH in the presence of concanaimycin B, a specific inhibitor of VETPs. So they saw that in freshly isolated disease, a similar alkaline phagosome pH found in the spleen, and in the they put disease control in disease with this inhibitor of VETPs, and when they inhibit the VETPs, the pH of disease go a lot higher, goes to the limits of the measure of the system of measure used. So in Figure 1e, they compare to bone marrow derived macrophage and see that it not replicates for them this alkaline situation. They put the same amount to disease, and the pH do not alkalinize and not sustains. The pH raises to not significantly and not sustains. and they put a high concentration, but the pH is not equally alkaline as disease. So, in conclusion, disease and not macrophages be an active machinery of phagosome alkalinization, which maintains the phagosome pH between 7 and 7.5 in the first few hours after phagocytosis. If the VTPs was inactivated, the phagosome pH in disease alkalinized strongly, showing that the VTPs is active at least to some extent in the C phagosomes. So next, they hypothesized that ROS generation by NOX2, the enzyme, remember, in the C phagosomes, could be responsible for the active alkalinization of phagosomes in disease. Since the intracellular localization of NOX2 transmembrane subunits in disease does not be shown, they analyzed the subcellular distribution of GP91-FOX, this big subunit here. They analyzed they did the disease defective for these subunits of NOX. So here we can see that GP91 FOX positive compartments are all around disease, including the dendrites. They localize to vesicular structures. They are distributed throughout the cell. So in figure 2b, they investigate the morphology of GP91 FOX positive structures, analyzing the intracellular stores of these site B558. That is all of these units. And they saw in their rows the structures, the membrane components of NOX, and they are next to the plasma membrane. And in figure C, they analyze the cells after 30 minutes of phagocytosis. This unity B558 was visible in phagosomes, indicating the recruitment of NOX2 to this compartment. To explore the recruitment NOX2 to phagosomes precisely, they purified the phagosomes from wild type and GP91 deficient disease after different time of phagocytosis. So as you can see, they do the iron western blot with wild type, with OK GP, and with defective GP. Defective GP OK does not show show nothing as expected, and they did a densitometry quantification of the western blots of GP FOX from wild type phagosomes, showing that membrane components of NOX2 are effectively and rapidly recruited to DC phagosomes. So the next attempt to determine if NOX2 was achieving DC phagosomes. So they saw that NOX2 is efficiently assembled and active in DC phagosomes, as you can see, compared to wild type to with GP91 subunits of NOX2 defective, the difference of fluorescence intensity, and here in wild type, in here with the defective, in wild type you can see it best, these units best than without the NOX2. And in the upper panels, it's a clear label of the phagosome membrane with anti-P47. The P47, it's this subunit here. And just a minute. and no labeling of the phagosomes was observed, consistent with the role of the GP in the membrane recruitment of P45 FOX. So we can conclude that NOX2 is effective assembled on the C phagosomes only when GP91 FOX is present, suggesting that NOX2 is functional in this compartment in disease. So we can disease have been shown to produce low levels of ROS. So next they went to see if NOS is efficiently assembled and active in DC phagosomes. So to address the possible generation of ROS, specifically in phagosomes, they covalently linked dihydrorhodamine 123, a dye that only emits fluorescence under oxidative conditions, to latex beads. In figure 3C, we can see the fluorescence microscopy, and 3D, we can see the FACS analysis. DHR coated beads become fluorescent after phagocytosis, showing the production of ROS in diseased phagocytes. So the inhibitor, and with that DPI, another inhibitor that I'm gonna say right now what it does. This fluorescent signal was a stronger inhibitor by this DPI, diphenylene iodonium, a specific inhibitor of flavin-containing enzymes such as NOX2. ROS production was not observed in phagosomes from this GP1, as I said, and you can see here too that we can see the difference between white type, showing the fluorescence, showing the assemble of NOX2, and the others. So we can conclude that the phagosome environment in disease is oxidative due to ROS generation by NOX2. And since like disease, macrophages have been shown to produce ROS during phagocytosis or after activation. To compare ROS production over time in disease in macrophages, they used conventional luminol-based techniques for ROS quantification. So they compare bone marrow derived macrophage to disease, and they did this quantification. You can see that disease sustained more high and sustained prolonged production of ROS than bone marrow derived. So disease display more efficiently and sustained in all three cases here NOX2 activity than bone marrow derived macrophages. So in figure 4A, you can see the ROS production in response to PMA was reproducible, high, and lasted longer. In figure 4B, you can see they evaluated the ROS production using the technique by covalently attaching luminol to latex beads. So the ROS production in DC phagosomes was stronger and sustained, and they used PMA macrophage after 2 hours of phagocytosis to exclude the possibility that bone marrow derived macrophage degrade luminol attached to the beads. So you can see that the hypothesis was excluded and disease were sustaining more ROS production. So in figure 4D, we can see the structures, the phagocytic structures of disease and bone marrow derived and BMMO, and after this time rate. So in figure 4D, after on this time here, the lower panels, after these times of phagocytosis, only the C maintained clear clear labeling of 447 Fox, these subunits, and you can conclude that sustained production of flaws in phagosomes from immature disease is due to a prolonged assemble and activation of NOX complex in DC phagosomes as compared to bone marrow-derived macrophages. We can see the structures here are similar. Here you can see they both sustain, but after more time, all disease sustained clear visualization of these structures. And this is linked with the NOX and the activity of the subunit P447, that is a subunit of NOX. So to test the possible involvement of NOX2 in the control of phagosome pH, they measured phagosome pH in disease with GP91 defective and wild-type disease. They saw that loss production in disease phagosomes cause alkalinization of the phagosomal lumen. We can see here that wild-type disease are more alkaline. The pH is 7.5, and it's less longer to go down and do not go down 7. You can see that defective disease does not sustain and acidify, showing that this unity of NOX is important to alkalinization of the medium. And here you can see the phagosome pH strongly decreased in the GP91 compared to wild-type. In figure C, we can see the contrast with the wild-type in GP91 bone marrow-derived macrophage. See, they do not sustain colonization, and this subunit is not crucial for macrophage as it is for disease to maintaining the alkalinization of the phagosome. And they did a blockade of the VTPs with the inhibitor, this inhibitor CON-B, and the marker alkalinization was observed in phagosomes from wild-type and not observing the defective disease, showing the NOX2 mediates phagosomes alkalinization. So here you can see the alkaline pH, the going to alkaline, are really alkaline, and defective are not as good to alkalinize. And here you can see the DP1, they blocked NOX2 with DPY in wild-type disease, and we can see the pH really go down, really acidify. But when they added CON-B, a blockade of ADPs, the pH got higher in 15 minutes. So if they inactivate the VTPs, the pH increase is showing that in the absence of NOX2, the VTPs still mediate acidification disease in DC phagosome. So here there's not NOX2, but when they inhibit the VTPs, they can increase a little the phagosome pH, showing that the VTPs has a role in the acidification of the phagosome pH. So we can conclude that NOX2 recruitment to DC phagosomes contributes to alkalinize the phagosome lumen and maintains the pH above 7 in spite of the activity of VTPs. In the absence of loss generation, DC phagosomes, So they next evaluate the functional consequence of such a drop in phagosomal pH in terms of antigen degradation. So to follow antigen degradation, antigen degradation sites in phagosomes, as opposed to endocytic compartments, they set up a quantitative cytofluorometric assay for phagosome degradation. After phagocytosis of beads covalently attached to the antigen ova, the cells were lysed and the amount of ova remaining on the beads were quantified using polyclonal ova-specific antibodies and FACS analysis on the isolated beads. So you can see figure C6a, a marked decrease over time in the amount of ova attached to the beads. Was observed in as compared to wild type. This decrease was less pronounced in the presence of protease inhibitors, leptin and peptine. So you can see this increase less pronounced. All in the phagosomes from cathepsin S deficient mice. You can see here. We can conclude that decreased immunofluorescence on the beads was due to degradation rather than the denaturation of ova. In figure 6c, we can see that can be induced a marked inhibition of ova degradation. A marked inhibition. Showing that the lysosomal proteases present in phagosomes are more active at the pH of 7.5 than 7.5 and 8. In this figure 6G, you can see that GP91 FOX deficient disease and the phagosomal pH is more acidic. The degradation of bead-bound ova was accelerated as compared to wild-type disease. Started really early than wild-type disease, showing the fluorescence intensity histogram. And you can see compared the intensity of beads. We can see that in wild-type it becomes more intact and it's consumed more in the disease that does not have the subunits of NOX2. So we can conclude that NOX2 achieved in the C phagosomes limits antigen degradation. So if it has NOX2, it goes slowly in degradation than if it does not have some subunits of the enzyme complex. So to examine if excessive acidification and antigen degradation in phagosomes affected antigen presentation, they evaluated cross-priming of adoptively transferred ova-specific CD8 OT1 cells into wild-type and GP deficient mice after selective targets of ova to resident disease. So we can see that wild-type has much more activation of this antigen than the defective. So you can see the activation was less efficient in the defective mice, suggesting that the efficiency of cross-presentation of this antigen ova by disease in vivo is decreased. The efficiency of cross-presentation of ova by disease is decreased in the absence of NOX. So they saw the percentage of phagocytosis in the beads dilutions. The wild type the wild type disease compared to defective disease, and the percentage having phagocytosed at least 1 lax bit. And you can see here the T-cell response. So they were trying to prove that this was linked to antigen degradation and not antigen presentation. So this graph is important to set up this more clear. So the presentation of the correspondence of peptide was similar in both DC populations. Sorry, I'm gonna go back here to present more clearly. The percentage of disease have phagocytosis disease failed to cross present always efficiently efficiently. The presentation of the correspondence of peptides already processed was similar in both disease populations, showing that disease from defective GP mice are fully competent for T-cell activation. So they are competent for T-cell activation, as you can see here, the peptides concentration. But the degradation is different. So if they degrade less in wild type, when they have NOX2, the presentation is best to T-cell. T-cell response is best. And so all these results indication that the effect in cross-presentation observed in GP deficient mice disease is related to antigen processing, this degradation, the level of degradation, and not antigen presentation per se. So next, you can see treatment of the disease with protease inhibitors that did not affect peptide presentation to the same T-cell hybrid. You can see in the right panel. Partial inhibition of proteolysis in GP defective disease overcome the cross-presentation defect, indicating that excessive antigen degradation in phagosomes from GP defective disease resulting from abnormal acidic pH is the cause of inefficient cross-presentation. So here they put the wild type disease, the defective disease, in the presence of these protease inhibitors. So the inhibitors, the inhibition of these proteases, it was less than the wild type. And here, just to get clear, let me see if I understand this graph. The treatment of disease with protease inhibitors gives this indication that the effective in cross-presentation is related to treatment of disease with protease inhibitors not affect the peptide presentation to the same T-side, not effective. So we have wild type disease, wild type with this inhibitor, defective disease with the inhibitor. You can see don't have much difference from the wild type. So the conclusion is that, I'm gonna say again, partial inhibition of proteolysis in GP defective disease overcomes the cross-presentation defect, indicating that excessive antigen degradation in phagosome from GP defective disease results from abnormal acidic pH is the cause of inefficient cross-presentation. So they close this case that that the excessive antigen degradation is the cause of the bad cross presentation. So if the DC degrade too much, they would not present, and it depends on the alkaline to not degrade too much. If it's acid pH, if the acid pH it degrades more than it should, and with proteases you can inhibit this degradation and put a less acidic and go best to the presentation. So as a conclusion, this article shows a novel mode of regulation of phagosome gasification and proteolysis in disease, a role for NOX2, a major player of innate immune response in neutrophils, and the regulation of adaptive immunity, genetic evidence supporting the idea that exacerbated antigen degradation is detrimental to antigen presentation to T cells, and identify one of the underlying mechanisms that limiting acidification in disease. So they saw that VATPs import protons across the phagosome membranes, and NOX2 cause proton consumption in the phagosome lumen. So when you have less protons in the phagosome lumen, the pH is more alkaline, and when you have more protons in the across the phagosome membrane, the VATP is a mediator, you have a more acidic alkaline. So this fine balance between the more protons and the mediator that put them and then pump them out, this pump of protons may make the difference in the presentation of antigens, because it's total as a professional presenter cell, totally depends on this delicate balance of not degrading too much and not degrading too little to make the best presentation, the best adaptivity immune response. So thank you. I hope you understand, and thank you.